Hello, good morning. Hope you guys had a restful weekend. Know you probably had a lot of evaluation last week to deal with, but you guys did quite well in the test for my course. And so that's the end of the first unit, and you won't be tested on those things again. Now we're going to begin our next unit. The second unit talks about the nervous system, and that's going to take us 2 lectures. So lecture 5 will focus on the cellular workings of the nervous system, how neurons communicate with each other, how signals are being transmitted. We're going to learn about all of that in lecture 5. Lecture 6 talks about more like the gross anatomy, what you call the different parts of the brain. Going to learn about the structure of the spinal cord, as well as different functions that are mapped to different regions of the brain. So that's lecture 6. Lecture 7 will be on sensory organs. So learning about the eyes, the ears, smell, taste, where do those information get processed in the brain, and just learning the different parts of those sensory organs. And the last lecture of the unit is the digestive system. So nervous system tends to be a little bit more involved in that there are a lot of terms that you have to learn. There are a lot of concepts that are a little bit obscure, if you would. And it might take you some time to fully digest all the material. But as usual, if you need help, please do let me know. Let me just enable the caption first so that if you need it, you will have it. Okay, let's get started then. All right, nervous system part 1. So when we talk about nervous tissue, there are actually 2 main components here. We have what's called the neurons, and these are the components that is responsible for generating and transmitting or conducting electrical signals. So they communicate with each other. They would send signals to different regions of the brain, to different regions of the body. And these information can be classified as sensory information in some cases. So they would have information that are, carrying information from like the eyes, from the ears. They contain sensory information, right? Sight and auditory. And then in some cases, they will be transmitting motor signals, which are responsible for telling a particular part of the body to move, or telling a gland to secrete some hormones. But regardless of the kind of information that it carries, the function of the neurons essentially boils down to transmitting, and processing signals. We're going to learn more about that later on. I'm going to map out the entire path for you from when you detect a change in the environment and how that change is being processed by your brain or sometimes by your spinal cord. And then we're going to have an appropriate action towards that change. So neurons are pretty precious in the sense that once you lose them, they're not going to come back anymore, right? You've heard people say neurons don't divide, and if they die off, then you're not going to get new ones. But if that's the case, then it's kind of puzzling, right? Because you hear people get brain cancer. How do you get cancer from nervous tissue if the neurons don't divide? Well, it turns out the cancer arises from the other group of cells in the nervous tissue, which we call the glial cells or neuroglia. So these glial cells, they are supporting cells for the neurons. Since the neurons are very precious, you want to protect them. You want to nourish them. You want to kind of pamper them. So that's what these glial cells are for, right? They outnumber the neurons 1 to 9 and takes up more than half of the volume of the brain. Okay, and they kind of huddle around these neurons and support them and nourish them. And in many cases, they form key components of our nervous system. So here are some examples of different types of glial cells. You don't have to know them at this point. We're going to introduce them slowly throughout the course. But sometimes you would have a specific type of glial cells for the central nervous system. And maybe we can just define what that is right now. Central nervous system is the brain plus the spinal cord. Okay, and everything else is called a peripheral nervous system. So sometimes you would have the glial cells that performs the same function, but one is found in the CNS, the other is found in the PNS, and they are named differently, even though they perform the same function. So we're going to learn more about that as well. So the brain cancer arises from these glial cells. Okay, these glial cells are able to divide, and when they are divide in an uncontrolled manner, then that gives rise to various forms of cancer. For example, astrocytes. People can get a form of cancer called astrocytoma, right? Ependymal cells, they could get ependymomas. Depending on which kind of glial cells is doing the uncontrolled division, they manifest as different forms of brain cancers. So that's just like an introduction to nervous tissue, right? We got the neurons who are doing the actual work, and then we are protecting the neurons with the glial cells. So let's take a look at what a typical neuron looks like, and for that we're gonna have to pull out the study guide, and the diagrams are usually located at the end. So there we go. That's a neuron right there, and we'll label that. Okay, so just gonna give you a moment to pull that out in case you don't readily have it available. Okay, so this is a typical neuron. Okay, do they all look like this? No, not necessarily. Okay, some of them has like 2 of these tails. So some of them look like there's a main body, and then they have 2 tails coming out. Okay, and some are shorter, some are longer. But this is like a typical one that you will find in a textbook when you are introduced to a neuron. So let's go through some of the major structures. Now, a neuron is a single cell. You probably should know this. So this is a neuron, okay? And it is a single cell. Okay, just by itself. And it would have all the features of a cell. So there's a cell membrane, right? Over here, you would see this circular structure in the middle. That would be the nucleus. And over here, that would be like a cytoplasm. And in the cytoplasm, you will find everything that you would expect to find in an animal cell. Mitochondria, endoplasmic reticulum, Golgi apparatus, and all the stuff that you learned last semester, they will all be here in the cytoplasm. Okay. So extending from the cell body, we have these tree branch looking stuff. Okay, so that is what we call dendrites. Okay, so the function of dendrites is to receive signals from another cell. Now, this other cell is usually another neuron. but not always. We're gonna, again, talk about that more later on. There's just so much information, it's impossible to, introduce everything at the same time. So, just gonna have some gaps here and there for now. And then as we progress through the course, through this lecture, we're gonna fill in those gaps. So dendrites, they will receive signals from another cell, and usually that other cell is another neuron, but it doesn't have to be. Now, remember, I told you to get some colors, right? So, for this class, this week is okay, but next week you definitely need to have some coloring pencils. We're gonna be doing a lot of coloring next week. Like, I'm just gonna show you the brain, right? Like, we're gonna be coloring this brain. We're kind of gonna be coloring these things, and then you're definitely gonna need to have some highlighters or crayons with it. Otherwise, it wouldn't make much sense. But for now, if you have a green marker, right, we can put some green arrows here. Okay, so these green arrows, they represent excitatory signals. Okay, so these are the signals that will tell the neuron to pass it on. Okay, it's like a go-ahead green light signal. It tells the neurons to transmit this information. And opposite to that would be inhibitory signals. The inhibitory signals are, as the name suggests, going to tell the neuron to, don't transmit this information. So at any given time, it's possible for a neuron to be receiving a bunch of excitatory as well as a bunch of inhibitory signals. And what happens at the cytoplasm in the body of the cell is they will add up all these signals and see which one is stronger. Right? If there are more excitatory signals than inhibitory signals, then the neuron will transmit. If there are more inhibitory signals than excitatory signals, then the neuron will not transmit. Okay, this is of course an oversimplification of what actually happens, but for the most part, right, it's just like voting. Okay, do you have more green signals or more red signals, and that determines whether it's going to be transmitted or not. Just going to pause there for a second to see if anyone want to ask anything.
Yeah, I just wanted to ask about the signals.
Mhm.
So the dendrites receive excitatory and inhibitory signals.
That's
Okay.
right. Yeah. So, I mean, there are some cases where we don't want to transmit things, right? Like a best example would be, if you're studying in the cafeteria or, in the food court or something, and then you're focusing on your work, right? Eventually you zone out the background noises, right? People chatting, in the next table, maybe music playing in the background, you eventually zone those out, right? So those are instances where the neurons are like, okay, we don't need those distraction right now. So we're going to block it out, right? And that's when you would have stronger inhibitory signals compared to the excitatory signals, okay? All right. So if we decide to transmit a signal, the signal is going to go down through this long tail here. Okay, so just going to highlight the middle here. That long tail, which might or might not split at the end, that is called the axon. Axon. And the purpose of the axon is just to allow the signal to get transmitted down the neuron. Okay, so transmit signal down the neuron. Now, a lot of neurons, they have these fatty substances that wraps around the axon. Okay, kind of like Twinkies. Okay? The filling is the axon, right? And then the Twinkie wraps around the fillings, right? These are called myelin. Okay, and the purpose of the myelin is to insulate the axon to increase transmission speed. Just like the wires, the copper wires in electronics in the house, they have to be insulated, right, by the plastic cover. That allows the signal to speed up. But the myelin does not cover the entire axon. Like, I mean, it does look like it covers the entire axon, right? But every now and then, you're gonna have a gap. You need to leave a little bit of a gap, and those gaps, they are called node of Renvier. Probably named after the person who discovered them or something. So the node of Renvier, they are gaps in the axon that do not have myelin. Okay. And as we will see, the function of the gaps is they allow ion exchange. Okay. So the signal that we keep on talking about is actually an electrical signal. Okay. And to transmit that electrical signal, we rely on ions that are being exchanged between the cell, cytoplasm, and the surrounding. And of course, you know the ions are charged themselves, right? So that's how they create the electrical signal. Okay, so the myelin will allow the speed to be faster. Last unit, we talked about autoimmune diseases, right? And I briefly mentioned multiple sclerosis. In multiple sclerosis, your immune system attacks the cells that makes the myelin. And so eventually the neuron is going to lose the myelin. And once that happens, signals will not be able to get passed down the neurons. And you slowly lose the ability to move certain parts of the body, for example. And if the neuron is responsible for connecting to your breathing muscle, let's say, right, and then you lose the myelins in those, then the person will have difficulty breathing, right? That's why it's a fatal condition to have. All right, so the signal gets passed down, and if there are myelins, then the signal will jump actually from gap to gap, from the node of Renvier to the node of Renvier, as opposed to get passed down, you know, in a linear fashion. And that jumping action, it's what speeds it up. Okay, so we can write down here signals jump from node to node. Okay. Not all neurons are going to have myelin. Sometimes you have naked ones. That's what they're actually called. Okay, when they don't have myelin, we call them naked. These naked neurons are usually quite short. And if they're really short, then the signal can just pass through them quickly without losing its strength, right? So there's no need to have myelin on those. Okay, but most of the longer ones in the body, you would need to have the myelin. So at the end, we have these terminal ends. Okay, we call them the axon terminal. And the function of the axon terminal is to send signal to another cell. And again, this other cell is usually another neuron. Okay, but it doesn't have to be. All right, that's it. for your neuron here? Any question about any other stuff on the page? Okay, great. Well, if you think of anything else, feel free to interrupt me later on. So we're just gonna learn some names. Let me move myself to the bottom left here. We're just gonna learn some names of how we call different things. I already told you earlier, right? CNS, the central nervous system, that's the brain and the spinal cord. Everything else that's outside of the brain and spinal cord, we call it the PNS. Okay. So if you take a look at my pencil here, that's like one axon. And if I have a bunch of them, running out of pencils here, give me a second. Right. There we go. So if I have a group of axons, okay, this bundle of axons, if you find this bundle in the PNS, like outside of the brain and the spinal cord, we would call it a nerve. Okay. You probably heard people say you struck a nerve, right? So a nerve is a bundle of axon. My pencils are invisible here. A bundle of axons in the PNS. The same bundle of axon, if it's found inside the brain or the spinal cord, we would now call it a tract. Okay, so tract and nerve are essentially the same thing. The only thing that's different is, where do we find it? Do we find it in the brain or spinal cord, in which case we call it a tract, or do we find it outside of the brain and the spinal cord, in which case we call it the nerve? Okay, it's kind of like how we speak differently in Canada than the US for certain words, right? Like after a meal here at the restaurant, we would ask for the bill, right? Okay, but in the US they would say, can I have the check? Check bill, same thing, but if you're in different places, you call them different things. So that's the same situation here. So there are a lot of these terms that as you will see when we learn the nervous system, that they essentially refer to the same thing, but we call them different things. So these axons are covered by myelin, right, for insulation to increase conduction speed. We know that already, right? So here you can see a bundle of axon. And they're grouped together to make it stronger, right? Rather than having just one single axon, it's very easy to break, and we don't want that. And we have various connective tissue that kind of binds them together nicely. And here is another example for the cells in the PNS. So if we're talking about the nerves, they are going to be myelinated by these glial cells called Schwann cells. And so Schwann cells forms the myelin in the PNS. You can see it here, right? And in the CNS, in the brain and the spinal cord, the oligodendrocyte is responsible for forming the myelin. Okay, so just two groups of different glial cells that, again, perform the same function. The function being creating the myelin. All right. So let's just do a quick poll to see if we are all on the same page. This should be pretty straightforward. Oh, except I just realized I need to teach you some of these terms as well. Sorry, Monday mornings. Okay, let's go back to here for a brief second, and let me introduce a few other terms to you. Okay, so I keep on saying, right, like the dendrites is going to receive a signal from another cell, usually another neuron, but not necessarily. So what is that other thing, right? Now I'm just going to keep it really brief here, and then I'm going to explain to you in a more detail later on. Okay, so for us to detect a change in the environment, whether like the external environment is getting colder or brighter light, right? Those are external changes, external stimulus, right? Or if we need to detect some kind of change in the body, we're gonna have to have some sort of receptors that are designed to detect those changes. Okay, so let's say we have the eyeballs. Okay, so this is the eyeball, and that would be an example of a sensory organ. We have other sensory organs, of course, right? The nose, the ear, the tongue, and so on and so forth, right? So that's gonna pick up like some change in environment. Maybe we turn on a lighter. Okay, so now we see light. Right? So that signal is going to get transmitted. Okay, but in this pathway, there are many neurons, right? It's like a game of a broken telephone. I don't know if all of you know that game, but you basically have a message from one person, and then you tell the message to the next person, and then the next person passes to the next person, and so on and so forth, right? So in this pathway, you will have neurons communicating with each other, right? So if I zoom in, I might have one neuron here, and then there will be another neuron here, right? And then the signal would go from this neuron number 1, all the way to the axon terminal, and then pass it to the dendrite of neuron number 2. And then the axon terminal of number 2 will pass it to the dendrite of number 3 and number 4, and so on and so forth, right? But what about the very first neuron here? Okay, that very first neuron gets the first-hand information, right? It didn't get the information from another neuron. It got it directly from the sensory organ, right? So that's what I mean, right? The dendrites usually will be receiving information from another neuron because it's part of a long chain. But if you are the first special one, you're the very first neuron in this whole pathway, then you'll be receiving it not from another neuron, but directly from the sensory organ. Does that make sense there? Okay, and then somewhere in the way, right, you might get the information to the central nervous system, and you're gonna process that information, and then you will send the signal back out to some structure. Okay, it could be a muscle, and maybe a muscle, right? Could be a gland. Okay, could be an organ, and it will tell these things to respond accordingly. Okay. Minas, is that like a primary? Yeah, like a primary source, right? So the sensory organ, the structure that is detecting the change, that's the primary source, right? So when that organ detects the change and it passes on to the first neuron, that first neuron, the dendrite of that first neuron is receiving signal from directly the sensory organ rather than another neuron. Okay. So change this to let's say a bright flashlight, okay? And let's say this is like in the middle of the night, 2 a.m. in the morning, someone decided to play a prank on you. They turn on the flashlight. So you open your eyes, the flashlight shines you, and then what is your response to that? Well, you're probably gonna block it, right, with your hand, right? So the muscle has to move in response to that bright light, right? So in this case, when the neuron gets to the very end, the last neuron in the chain, rather than passing the signal to another neuron, is going to pass the signal to a muscle, right? That's what I mean here, right? The axon terminal sends signals to another cell, but not always a neuron, because if you are the last one in the chain, you will pass it to a muscle, right? You might get a little bit scared because you're wondering, well, why is someone shining a light at my face when I'm sleeping? So your heart rate might go up. Right? So the organ is also targeted, right? You might secrete a little bit of adrenaline, right? You don't know what's going on, right? Do I have to fight this person who's shining the light at me? Do I have to run away? So that adrenaline gets you pumping, right? So all these end targets that are going to be responding to the stimulus, we call these the effector. Okay, effector. It has nothing to do with the effector that we learned in last unit. Okay, these are in completely different contexts. Okay, and just one last term, and again we will revisit these things later on. When the neuron communicates with another neuron, where they communicate, that's called a synapse. Synapse. Okay. We'll have a whole 20 minute session on synapse later. But for now, it's just where the two neurons are very close to each other. So that's a like a really brief overview. Any question at all? How fast does all this happen? Pretty fast. Okay. You never had a like when you're older, when you're younger, you never had this situation where like your older sibling or cousins like try to scare you in the middle of the night or something, or shine a light at you, or someone flip on the light, right? You react almost instantly, right? Okay. Think about my kids leave toys on the floor all the time, right? If I step on one of those Lego pieces, I jump right away, right? It's a reflex, right? So this is a really fast transmission. Okay. Less than a second. And we want that, right? Because if you're driving, you want your reflex to be fast, right? You see a rabbit crossing the street, you want to be able to see the rabbit, process the information and say, I don't want to run over the rabbit, and then send the signal to your legs to step on the brake, right? All that happens in split second, as they say. I'm just answering someone who private message me, asking like how fast all this happened, in case you're wondering why I was talking about that. Are we good? Sorry, I'm a little bit sluggish this morning. It'll get better. I'm just warming up right here. Okay, so that is that, and we were talking about this question, right? Okay. So you probably know the answer already. A signal is transmitted from one neuron to another through which of these? Let's start the poll again. So based on everything I told you just now, how does the signal go from one neuron to another? What do we call that location when you are jumping from one neuron to another neuron? Okay, not how the signal is passed down the neuron, but how does it go from one neuron to another neuron? What do we call that location? Okay, and the correct answer is the synapse. Okay, synapse. The axon is how the signal gets passed down, and then when it goes from one neuron to another neuron, that location is called a synapse. Here's another one. You probably could look it up in the slide, but use your memory, see if you can recall that information. Yeah, amazing. Everybody get it right. That's the oligodendrocyte. Okay. Sometimes it's called myelin sheaves, like extra word, but we can just call it myelin. It means the same thing. Okay, flow of information in nervous system. This is a long note. Okay. It's a elaboration on the whole eyeball thing that I was talking about. So let's go back to our study guide here, and we will draw it out. Okay, so this is the first page. You can draw it on a separate piece of paper, okay, and then make it look nice later on. So let's say we have a stimulus. Okay, so again, maybe let's do a little candle here. Okay, and then you accidentally put your hand on it without knowing. Okay. So if you put your hand on something hot, something sharp, immediately, right? You're gonna remove your hand, right? So what happens between touching the hot surface and then letting go of your hand? a bunch of things happens in between, right? So let's take a look at that. So first of all, like I said, we're gonna need to have some kind of receptor that are dedicated for detecting these things. And we're gonna learn about all these special receptors in lecture 7. Okay, but suffice to say, we have receptors that are dedicated for detecting light. We have receptors that are dedicated for detecting sound. We have receptors that are dedicated to detecting temperature change. Okay, and these are embedded all over the body in your skin. So over here, we're gonna have a sensory receptor for detecting the change in temperature. And we're gonna represent it like this. They do kind of look like that. You just have to kind of trust me. This is a receptor, sensory receptor. Okay. And this particular type of sensory receptor is sensitive to change in temperature. Okay, we call it a thermoreceptor. Let me just maybe put it on top here instead. So the sensory receptor will detect the heat from the flame, and then we are going to send that information through a sensory pathway to our central nervous system. Okay, so this blue path represents the sensory pathway. Okay, we call it the afferent pathway. It is made up of sensory neuron. And, sorry, give me more space. And it carries sensory information towards the CNS, central nervous system. In this particular example, the sensory information that is being carried is related to temperature. But it's the same thing for all other sensory structure, right? If we're talking about the eyeball, then the sensory information would be sight. We can have sensory information about sound, about taste, about what other things can you feel with your hands, okay, other than temperature? And what are some other other things that we can we can feel? What are some other descriptor?
Pressure?
Yeah, so pressure, that's another sensory information. Anything else? Texture, exactly, right? Is it rough? Is it coarse? Is it smooth, right? And then maybe one more. Pressure,
Pain?
texture, pain, exactly, right? Pain, yeah. So you know, if you touch something sharp, if you get, cut your fingers, right? Pain would be sensory information as well. Okay, so all these sensory information is going to be traveling from the sensory receptor through the afferent pathway towards your central nervous system. Okay, and this path here is made up of the sensory neuron. Okay. Now, once we get to the central nervous system, we're going to have another neuron here. Okay, and this one actually has 2 axons. Okay, so this is what we call an interneuron. Interneuron are only found in CNS, only found within the brain or the spinal cord. And their function is to process sensory input and determine appropriate motor output. Okay, like what are you gonna do about it, right? And sometimes the appropriate response is to not respond. Okay, let's say I don't know. I don't really have a good example on top of my head. Like, you know, sometimes they say if you run into a bear in the wild, right? Like the best response is not to run away, right? Because if you run away, the bear is going to chase you. You're supposed to stay still and hope they don't notice you, right? So, you know, in the case of that, the motor response would be, you know, to just stay put. Okay. So here I'm gonna draw an imaginary boundary here. Okay. You know, I'm going to cut through my own words here. And this would be the CNS, the central nervous system, brain plus spinal cord. And in this case, your spinal cord is fully capable of making that decision for you. Okay, it would take too long for the information to go up to the brain and then make a decision and come back down. Right? That's like a second too long, and you might, you know, burn yourself more than you need to. So that's why the spinal cord in this case will help you make the decision. Okay. So I hope you know what the appropriate motor response is in this case, right? Would be to remove your hand. So then you're gonna have another pathway that goes back to the hand and remove it. Okay, so this one is called the efferent pathway. Okay, so efferent from PNS towards CNS. Okay, this is PNS, everything outside of the brain and spinal cord. And efferent is the other way around. This is made up of motor neuron. And it carries motor information towards what? Can someone fill this in for me? I'm looking for a word that we learned today. What is it going towards? This motor information. Sensory organ. Okay, so that's good try. Not necessarily though. It doesn't have to go back to the hand, or it could go to another part of the body. Anyone else wants to try?
Peripheral nervous system? Or
Oh, yeah, it's going to be traveling away from the CNS into the PNS, but ultimately, yes, Min, you got it right. It's going to be going towards the effector, right? Effector. Remember, the effector is the final destination of this pathway, right? It's the final thing that's going to carry out the motor output, right? So in this case, the effector happens to be the same as where the sensory receptor is, right? So I understand why, you know, some of you might think that's the final destination, but it doesn't have to be, right? In another situation, like the one with the flashlight, right, shining at your face, the heart is the effector, right? It causes your heart rate to go up, right? So they don't necessarily have to be the same thing. So that is a complete pathway. Do we have any question about this?
Is the efferent pathway made up of many motor neurons?
Yeah, they're like a bunch of them grouped together. Remember this picture that we had, right? Like this is a bundle of axon, right? So it's a whole bunch of neurons forming that particular pathway, right? If you just have one neuron, then it's like super delicate, right? Like, you know, if I break my arm, I might break the nerve completely as well, right? So that's why we're gonna have a group of them to make it stronger. That actually makes up a good point. So this here, yeah, sorry, go ahead.
I'm only asking that because it's worded made up of motor neuron.
Oh, sorry. Yeah, motor neurons. There we go. Is that better? Thank you. Okay,
Yes.
so this whole thing, this whole thing right here, I don't know if I should talk about now, but it might confuse somebody, but I'm gonna write it down now. Okay, we're gonna come back to it later. This makes up like a nerve, right? A nerve. Okay, so you have a, you know what? Actually, I could do it separately. This would be like a motor nerve. Okay, and this over here would be like sensory nerve, right? Okay, because it's a bunch of axon that are found outside of the CNS, right? So if you damage your sensory nerve, you won't be able to feel a particular sensation. You can still move it. If you damage your motor nerve, then you won't be able to move, right? You guys probably are familiar with ALS, right? You guys know Stephen Hawking, right? He had ALS. He couldn't move the body, and that's an autoimmune disease that results in degeneration of the motor nerve, right? Motor neurons, right? And if you lose that part of the nervous system, you won't be able to move. Okay, let's just summarize it here with all these fill in the blanks. It's basically what I wrote up there already, but it's always nice to write it out again. So this is the which path is this? The sensory input. Is it the afferent path or efferent path? You should think about that. And hopefully you are thinking about afferent. So afferent pathway is made up of sensory neurons. You can see it's plural here. That carry sensory information towards the central nervous system. Within the central nervous system, again, I have to stress that it could either be the spinal cord alone or the brain or a combination of both. That's where integration occurs. Okay, so integration basically involves interneurons, which are found inside the CNS only. And their job is to process sensory inputs and determine appropriate motor outputs. And then for the motor output, we have the efferent. Some people pronounce it efferent as well. Same thing. So the efferent pathway is made up of motor neurons that carries motor information towards the effector. And just to reiterate, effector can be a muscle, could be a gland, could be an organ. It's just whatever is carrying out the response. All right. That's that. So we good with this? We'll talk about this like one more time soon. Yeah. Go ahead. Sorry, Thashana.
Can you just go back to the notes that you made for
Sure,
me?
sure.
Yeah, the part about Efferent. Thank you.
Yeah. Let me know when you're done with it, okay? We can move on. It will come together in the end, okay? I promise. Right now, it's just like pieces of information here and there. Okay, I have another private message. If you have slow reaction time, does that mean the signal are slow or damaged? That's an interesting question. Not necessarily damage, okay? Like some people are just wired differently, right? Their synaptic pathways is wired differently, and that result in slower transmission time. Okay, and sometimes the processing power is different for different people as well, right? Like take multiplication table, right? Some people can recite it very quickly. Right? 2 times 2 times 6 is 12, and then some people can take a longer time to recall that information. And sometimes reflexes are kind of like that, right? Because of the wiring of the pathways that are done in the brain, it causes certain people to have better reaction time than others. But these are trainable traits. Okay, like if you keep on practicing on it, right, you get better at it, right? So if you're really slow at think of like an athlete, right? Okay, they have to be very fast in reaction time if they're playing, I don't know, like hockey or something, right? And of course they're not naturally born with those kind of reflection and reflexes, right? Think about like the goalie, right? They can train, right, so that they respond faster over time. Lisa has an
Okay, Professor, thank you.
okay, yeah, thank you. And Lisa has another question. The effector here is the muscle from our hand, right? Yeah. But yeah, yes and no. I mean, technically, to remove your hand, if you touch a hot surface, to remove your hand is actually muscle in the arm, right? Okay, but like close enough. Yeah. I hope that answers your question. Okay. Another thing I want to mention is the way I present this to you is like a linear path, right? Like it starts here and then it ends up back in the hand, right? But don't forget, if it's something more serious than like a candle, like if you actually cut your finger or something and it's like a quite deep cut and it's bleeding all over, right? Not only you recoil your hand, but like your heart rate probably goes up a little bit as well, right? So sometimes these paths can split, right? And then it will go towards another effector. Okay, like it might go to your heart and cause your heart rate to go up. So it's not always just linear, right? It's a very complex system. But to break it down into a simple circuit, it helps us understand a little bit more. Okay. What else do we have next? Let's see. Yep, so this is just another example, right? Like I was talking about like the bunny crossing the road, right? But another another more relatable situation, perhaps, if you drive is, you're driving on the freeway and then all of a sudden you see a cop, right, pointing a radar gun at you, and you're like, oh no, I'm driving too fast, right? So in that instance, you see the cop, right, and then you process the information. You're like, oh no, I'm going too fast. I'm gonna get a ticket. And then the appropriate response, right, would be to cause the calf muscle to step on the brakes, right? And if you are familiar with the situation that I just described, like you had that experience before, your heart is gonna be, jumping out of your chest as well, right? Oh, I hope he didn't see me, right? So this is another classic example of how the pathway will split. Not only will it go to your leg muscle to press on the brake, but it will also go to maybe your adrenal gland, release a little bit of adrenaline, goes to your heart, cause it to increase the heart rate a little bit as well. So it's like a multi output system. And also, same as the sensory input, right? Is not just the eye necessarily, right? There are other things that you pick up, right? You pick up sound at the same time, right? And your brain integrate all that information and then decide what the appropriate response is. Okay, but it's just being presented this way because it's easier to understand. Oh boy, another note. All right, let's do this. What happens when there's a serious injury? Injury to where? Can you elaborate a little bit, Min? Oh yeah, okay. Sight is numb. Yeah, we're gonna get to that later. Just hold that thought. We will eventually talk about that. All right, so now we're gonna do a what is this? This is nervous system, part 1, class notes. We have a lot of notes to take for this lecture, okay? So make sure you have a lot of papers and organize them. Okay, so division of nervous system? Of nervous system. Let me just take a peek at what's coming up next so I don't. Okay. We're good. So we have the nervous system. By the way, if this is your paper, right, you probably want to write nervous system right here in the middle, okay? Not on the side, okay, in the middle. So nervous system. Okay, so we have our glial cells, we have our neurons, but we are talking about functional division here, okay? Like different departments responsible for different things. Okay, so what departments are there in the nervous system? Well, we already kind of know, right? We have the two main division. Do you know which two I'm talking about? I've been mentioning them like all class long. Which two divisions do we have?
Central and peripheral.
That's right, the CNS and the PNS. Let's just write them out. Central nervous system. And you're probably sick of hearing me say this, right? Like brain and spinal cord. That's what they include. We all know where the brain is, right? But the spinal cord, it runs basically like from the base of the neck all the way down to close to where the butt is. And everything outside of that, that would be the PNS, peripheral nervous system, which is PNS. Okay. So just really rough diagram here to help you understand what's going on. This would be your brain. Looking at it from the back, for example, from the back of a person. And this would be like the brain stem. Remember we learned about the brain stem in lecture 4, medulla oblongata, that's part of it. And then it's going to transition into the spinal cord and it's going to come down all the way. Okay, so this is brain. And then this is spinal cord. Did you know the spinal cord becomes like a ponytail at the end? Okay, it opens up and literally becomes ponytail looking. Okay, so this is the spinal cord. So that's CNS. Now coming out of the brain, we would have these bundles of axons. And what do we call bundles of axons again? Do you guys remember? Bundles of axons. If it's inside the brain and spinal cord, yes, we call them tracts. But now it's outside, right? It's not part of the brain anymore, right? It's coming out of the brain. So we call them nerves. That's right. So these are cranial nerves. Cranial nerves. Okay, and we have 12 pairs of them. Okay, cranial nerves comes out of the brain. They connect directly to the brain. Okay, and then we have probably I should use a different color. We have these ones that are coming out of the spinal cord. This looks like some kind of creature from like a sci-fi horror movie. These are the spinal nerves. And you have 31 pairs. Okay, the way I remember is, there's 12 months in a year, and then 31 days in, most months. So 12 pairs of cranial nerves and 31 pairs of spinal nerves. Okay, so this spinal nerve, right? Try to stay with me here. This spinal nerve, if I look into it, I would find the sensory nerve and the motor nerve, right? In the same bundle. It's like your telephone cable. Right? Cut open it, you will see the incoming line and the outgoing line, right? So within this spinal nerve, you would see the motor and the sensory. Okay. So just to recap very quickly for the last time. The black stuff here, brain and spinal cord, that's the CNS. The blue stuff and the green stuff, everything that's out of the brain and the spinal cord, that's part of your PNS. Okay, and these are your motor neurons, these are your sensory neurons. And depending on what kind of information they are transmitting, we categorize them into different divisions. Okay, there are different departments, right? I keep on saying. So let's take a closer look at what these departments are. So your PNS is separated into somatic nervous system. Okay, SNS, and autonomic nervous system. ANS. Just to keep it simple, basically, somatic nervous system deals with transmitting signals to and from body parts that you generally have voluntary control over. Okay? So transmit signals, transmit and process, we'll say transmit and process, transmits, processes, is that a word? Information to and from parts of the body that we have voluntary control over. Okay. Now, what does that actually mean, right? In practical sense. Well, remember that example we did with the candle, right? Touching the hot candle. The hand is where you touch the hot candle. The hand is something we have voluntary control over, right? So, you know, if I'm gonna map that out for you, right, it would start right here, okay, and is going to go towards the central nervous system like this. Okay, so this is the burning the hand, right? Burning hand. And then the interneuron is going to process that information, and then it's going to send the signal back out to the somatic nervous system, right? Okay, to remove the hand. That's the motor response. Okay, because it originates and end in a part of the body that we have voluntary control over. That's the job of the SNS. Okay. Now, for ANS, it would be the same thing, but you can kind of guess what the description would be, right? So this transmits and processes information to and from parts of the body that we have no voluntary control. Okay. So an example, instead of like a burning hand, could be like kidney stones, let's say. Okay, so kidney stone is going to create quite a bit of pain, right? And that pain, that sensory information is going to be transmitted through the ANS here. But it's the same path in terms of, it starts here and then it's going to go towards your central nervous system. But this time is like I said, right, like kidney stone, pain. And then the response could be SNS. Right, like if you think about it, you have pain, and then like you might you can't really see me, but like you might touch that part of the body and be like, oh, that hurts, right? So touching that part of the body to kind of soothe the pain, that's processed through the somatic system, right? Because then the hand is something that you have voluntary control over, right? Do you understand? Like it all depends on whether the destination or the source is from something we have control or something we don't have control. Do you guys have any question?
Professor, I'm a little bit confused.
Sure.
Like for the autonomic nervous system, like is it related to our organs, like inside our body, so we don't have control over?
Yeah, for the most part, right? So if the source of the senses that we're talking about, right, originates from the inside of the body, then it's going to be conveyed through the ANS.
Oh, okay, thank you.
Another example is like heartburn, right? You have a heartburn, you ate too much, your stomach hurts, right? That's going to be again ANS, right? But in terms of what you do, the response to that might or might not be ANS. It could be SNS, right? Just rubbing your tummy, for example, right? Sometimes the response is also ANS, right? Like, for example, your blood pressure drops, and we talk about we need to squeeze the blood vessels, right? Vasoconstriction, right? So in that case, both the input and the output is going to be carried out the ANS. Does that make sense? Bruce, it's like when your oxygen level is low, you breathe faster and deep. Yeah, exactly, right? That would be another example of where the source, this input, is ANS, and then the output, I would say, is a little bit of a combination of both, right? Because breathing muscle is something that you can voluntarily control, right? So acid in the ANS and pain is in the SNS. Okay, sorry, can you clarify that or rephrase it? The question. Just want to make sure that I understand. Are you saying like is pain always originating from SNS? A heartburn, okay. So it's not so much about the acid or the pain itself, but the fact that the pain is happening in an organ that we have no voluntary control over. That's why it is the ANS. Does that make sense? Okay. I'm not gonna really be tricky on the quiz to ask you to distinguish, like, whether this is SNS or ANS, right? The bottom line is you have to think about, is this from or to something that we have control over or not? If it is, then it's SNS. If it's not, then it's ANS. Okay. Even pain can be SNS source or ANS source, right? Okay. If you fall and bruise your arm, is that SNS or ANS? You fall down, you scrape your arm, scrape your knees. Do you think that kind of pain is transmitted through the SNS or ANS?
SNS?
Yeah, because like your arms and legs, you normally have voluntary control over, right? Okay. And if someone is having a heart attack, the pain, right, that's ANS, right? Okay. Enough of that. Now this kind of signal, this kind of input through the ANS, instead of calling it a somatic input, This is called a visceral input. Okay, visceral source. Visceral source. Okay, as opposed to like somatic source. Okay, somatic means like bodily. Visceral is usually referred to things that are deeper in the body. Anyone confused about these lines, these blue lines and pink lines? They're just examples that shows you, which way information gets transmitted, right? I hope they're not too confusing. Okay, just one more. The ANS has an additional 2 subdivisions. And we have the sympathetic nervous system. And we have the parasympathetic nervous system. These 2 divisions, they are antagonistic to each other. That means they work against each other. They work opposite to each other. Okay? We'll talk more about it in the PowerPoint slides, but for now, the sympathetic system is most active for like fight or flight. When you're in danger, when you are in a stressful situation, and sometimes when you are in an excited state, the sympathetic system is most active. Whereas the parasympathetic system is more like rest and digest, they call it. This is more a relaxed mode. Okay. So if you think about fight or flight, your heart rate goes up, right? Rest and digest, your heart rate goes down. So both of them is going to have the same effector. They both target the heart. One will cause it to go faster, one will cause it to go slower. And they kind of always goes against each other. And depending which one is stronger, your heart rate will either go up or go down. It's never exclusively one form or the other. It's just, in certain situations, depending on what you are experiencing, one division is stronger than the other. And these things are part of your ANS. Okay, so there you have it. That's the entire division of the nervous system that we're going to be learning. Okay, I think we should take a break right now. We'll resume at 9:50. Go through these diagrams that we just drew. See if you have any questions for me afterwards. Okay, see you in a bit.
So, Professor,
Yeah.
Which one of the information here is on the central nervous system?
The central nervous. You mean which division is in the central nervous system?
Information for the part that transmit and process information? That's the ANS, no?
I see. Okay, so that's a good question. The ANS and the SNS will ultimately send all those information to the CNS. Okay, where the ultimate decision is going to be made. Okay, so this is like the final destination of all sensory information. Okay, and then you will make the decision of what to do with it in the CNS before sending it out again. Okay.
Okay, thank you.
All right. Okay, see you guys in 5 minutes. Hello, let's keep going. Let's go through the PowerPoints and see if we can clarify things a little bit. I do understand this is like very confusing stuff, and it does take some time to fully understand it. But you know, let's keep going and see if it gets any better. So this is what we were talking about before the break, right? We have the brain, we have the spinal cord, and coming out of the brain, we have all these cranial nerves, right? 12 pairs of them. And then we have the spinal nerves. And depending on where it comes out from the spinal cord, we call them different names. You don't have to learn the names. You just have to know that there are 12 pairs of cranial and 31 pairs of spinal nerves. And in fact, each of the 12 pairs of spinal nerves, they have dedicated function. Right? So the first pair, we call them olfactory cranial nerve, which is dedicated for smell, right? And if it's red, shown in red, it means is purely for sensory purposes. Okay? We have some that are mixed. Right? For example, the vagus cranial nerve, the pair number 10, it has both motor and sensory information. Okay? So if you see motor, it goes to the heart, lungs, bronchi, sensory, also lung, hearts, bronchi. And if you think about what we said with the division of the nervous system, right, these nerves would be part of the ANS system, because these are things that we don't have voluntary control over, right? Okay, so that's how we categorize them, right, in terms of, you know, functional division. So you don't have to memorize these cranial nerves, but you know, if you become a nurse later on, you probably have to learn this in your practical nursing program or nursing program, because to assess a patient for damage of the cranial nerves, there are different tests that you have to do, right? There are specific tests that you can do to see if the person has damage to their trigeminal nerve, for example, right? And you will learn all that in your next program, probably. So the ANS is divided into the sympathetic and parasympathetic system, we said, right? And they exert antagonistic effects on the same organ. It goes to the heart, like I said, the sympathetic system will speed it up, whereas the parasympathetic system will slow it down. In terms of like the lungs, right, it would increase and dilate the airway so that you can intake more air. Remember, you are stressed out when the sympathetic system is activated. Right? You're going to need to have more air, more oxygen to sustain that state. Whereas if you're relaxing, then it's going to constrict your bronchioles, right? So basically, the sympathetic system is most active during emergency situations, okay, where the person is required to fight the adversary or run away from it, right? Due to my experience with getting bitten by a dog, I am truly afraid of dogs nowadays. Okay, like if I see a dog and we're walking, like I want to stay away from it. So if an angry dog like that is barking at me, I am gonna, you know, uncontrollably have my sympathetic system switched on. Okay, so I will have increased heart rate, dilate the bronchi, dilate the pupil, so I can see better, right? We're gonna have increased energy resources in case I need to run away from the dog, right? And at that moment is not the right time to think about digesting your food. So the sympathetic system is gonna inhibit the digestive tract. Okay. Interestingly, when you are like on a roller coaster or when you're super excited about something, your sympathetic system is also turned on. So, you know, imagine the last time you were on a roller coaster, you probably experienced some of these things as well. Now, the sympathetic system can sometimes get turned on in a situation where you're not in danger, but like stressful. Okay, I've seen this happen on tests. Okay, sometimes students get very nervous when they write my test. I don't know why. My test, I would argue, is reasonably fair. Okay, but sometimes people get really stressed out. And one time I had a student who was so stressed out that they barfed, and then they're like, "Oh, I'm very very sick. I can't do this." And that's because the sympathetic system causes severe inhibition of the digestive tract. Right? Now is not time to think about the digestion. We don't have time. We have to dedicate resources for all these other things, and then your body empties whatever is in your stomach, basically. Okay. I also had a similar experience when this was like, I don't know, like 15 years ago, maybe. I had my first interview to be a teacher. This was at a high school, so I applied to be a high school teacher, and I got the interview. Okay, but for some reason, I was so nervous at the interview that my mouth ran dry and I couldn't talk properly. Keep on, like, coughing and nervously drinking water, right? And that's the sympathetic system, again, inhibiting digestion, right? Salivary secretion, secreting saliva is part of digestion, right? And when you're stressed out, your body tends to shut that down. I did not get the job, but that's okay. I got this job eventually. I had to mentally prepare myself not to activate a sympathetic system for this interview, and I think I did okay. On the other hand, the parasympathetic system is the rest and digest. This is when you're just, very relaxed, okay? Maybe doing some yoga, maybe chilling on the couch, watching some TV, right? Then now it's time to do housekeeping things for the body, right? You want to slow down the heart rate. You want to do some healing, maybe, right? If your body has some kind of injury that it needs to heal, right? The parasympathetic system promotes that kind of stuff. Your pupil gets smaller, less light enter your eyes, so that maybe you're getting ready for bed. And now is definitely the time to digest your food and just let the body carry out its normal tasks. Yeah, we don't want to have the parasympathetic system dominating all the time. Right? We need to maintain mental alertness of what's going on around us, right? So we need to have the sympathetic system to be in place. We don't want this to be overly active all the time either, right? Because then we're gonna be like so worked up, right? Very stressful all the time, and then that's not good for the body. So it's not about whether sympathetic is better or parasympathetic is better. It's all like dependent on the situation, right? And it's a balance between the two. Okay, so like on the quiz, you probably will expect some questions where it asks which of the following is promoted by the sympathetic system. So rather than memorizing a bunch of things that is controlled by the sympathetic system, just think about how you feel when you're in a stressful situation, right? Well, of course, my heart rate's going to go up, right? You're going to need more air, need more light, need more energy. That will help you, answer the questions. What about like sweaty palms and sweaty hands? Right? Do you think that's sympathetic system or parasympathetic system?
Sympathetic system.
Yeah, probably sympathetic, right? Like you're getting all nervous and all sweaty hands. Okay, that's it. All right. This is a multi-select question, more than one correct answer. Looks pretty straightforward, but I want you to think carefully, okay, to see which of the following is or are part of the PNS. Okay, thank you for answering. The cranial nerve, that's definitely PNS. This is coming out of the brain. It's not part of the brain. Spinal cord is CNS. Brain is CNS. Sympathetic system, right? Spinal nerves, right? So all these are PNS. Basically, anything that's not brain and spinal cord, you can safely say that they are PNS. Okay, here's another question. Yeah, very good. Okay, PNS is divided into ANS for involuntary stuff and SNS for voluntary parts of the body. Very good. Okay, more notes. This note is really, really long. Okay, you can see our PowerPoints are almost like running towards the end, so it's just gonna be taking notes for the rest of the class. All right. This next part is really confusing, I have to say. All right. And so if I'm not making sense, if anything is confusing, please just stop me and ask anytime. So we are going to be taking a note on something called action potential. And what does that mean? Action potential means basically this is how I'm just going to write in plain English. Okay, this is how signal This is how signals are transmitted down an axon. So we know this is our neuron, and then we have another neuron here. Okay. So the action potential is talking about how signal goes down the axon like that. Right? And remember, this is the synapse, right? Okay. We probably will not have time for that today. Next class, we'll talk how the signal goes from one neuron to another neuron. But for today, we will talk about how the signal is passed down the axon. Okay. I would suggest you have two pieces of paper. Okay. One piece of paper, we're going to draw a bunch of diagrams and make some notes, and then another piece we're going to draw a graph. Okay. And you're going to have to have both things here for it to make sense. That's just my recommendation. Let's start with some background information. Try not to look this up on the internet and watch videos online, because we try to make it a little bit simpler here. Okay, if you watch online, I worry you got extra information and it's gonna just confuse you. Right? If I'm gonna try to post some videos for you that I made to help you understand this, but just try to stay with what I give you. That's what I'm saying. So background information. Let's talk about the sodium potassium pump. Now, if you were in my class, we talked about the sodium potassium pump in lecture 4. What about students from either Miran's class or TJ's class? Did you guys learn about the sodium potassium pump?
Yeah, we learned that.
Yeah. Okay. Great. Good. So the sodium potassium pump plays an important role in how neuron functions. Okay, so just to quickly recap, it uses ATP to pump 3 sodium out of the cell in exchange for 2 potassium into the cell. So we're gonna take our neuron again, and then I'm gonna Focus on the axon. Okay, so this is the axon. And located along the axon membrane, we're gonna have a bunch of sodium-potassium pump. Okay, and I want to represent them with these purple ovals. I'm just gonna draw a few of them. Okay, so that's the sodium-potassium pump. Like we said, the job of sodium-potassium pump is to pump out 3 sodium in exchange for 2 potassium. Okay. Now just gonna put some labels here to help you orient yourself. This is the cytoplasm, like inside the axon, right? And then this would be the outside, the extracellular fluid, ECF, outside. I hope you're doing this in color, huh? Otherwise it's very difficult to make sense of it all. So the sodium-potassium pump is pumping sodium out, which means on the inside, we're not gonna have too much sodium, right? But on the outside, we will have more sodium. Same thing for the top, but just gonna keep it simple like this. So relatively speaking, on the inside of the cell and in the cytoplasm, we will have low sodium, low concentration of sodium. That's what the square bracket means. And then on the outside, we would have high concentration of sodium. I'm just gonna pause for a second and let you catch up. Give me a thumbs up if you could when you are ready to move on. Okay. Just gonna wait a little bit as people copy them down. Okay. Now, similarly, for the potassium, because the sodium-potassium pump keeps on pumping them in, we will have more potassium on the inside compared to outside, right? So if I have to write a descriptor, we would have high potassium on the inside, relatively speaking, right? Compared to outside. Now, I want you to think about this, right? When given the opportunity to do so, do you think the sodium would move into the cytoplasm or out of the cytoplasm? Let me repeat the question. Ignore the sodium potassium pump for a second, okay? Just ignore all these purple ovals. When given a chance, do you think the red dots, the sodium, would want to come into the cytoplasm or out of the cytoplasm? Let's do a vote. so I know what you think. If you think it's going to come into the cytoplasm, please choose A. Out of the cytoplasm, please choose B. Oh, okay, good. Overwhelmingly say, they're going to come in, and you would be correct, because things naturally wants to go from high concentration to low concentration, right? Okay, so the sodium really wants to come in, but they can't do it because they are charged. And you might remember, like, the membrane does not allow charged particle to go through, right? Similarly, when given the opportunity, the blue dots, the potassium, they would want to leave, right? Make sense? Because things naturally wants to go down their concentration gradient from high to low. Okay, so that's something that you should know. Should I write it down? Yeah, I guess I'll write down. Okay, so under the right conditions, Na will move from high concentration to low concentration, right? Into the cytoplasm. Similarly, you can write the same for potassium. Okay, but it will be out of the cytoplasm. So again, under the right condition, K+ will move from high concentration to low concentration out of the cytoplasm. Min asks if this is diffusion. Not quite, and you will know why soon. Okay, just again, hang on to that thought for a second. Any questions so far? You have to understand that. Otherwise, like, the rest is gonna be like super confusing for you afterwards. You understand they cannot do that now, right? Because the membrane prevents the charged particle from passing through them, right? And you have the hydrophobic layer, right? But they really want to come in, the sodium. They just can't yet. And then the potassium, the blue stuff, they really want to go out, but they can't. Okay, that's why this is a pump, right? ATP, uses ATP because you're pumping the sodium out against the concentration gradient. Similarly, you're pumping the potassium in against the concentration gradient. Okay, this next part, I really need you to think about it. All right, you don't have to tell me. You can just, as I describe it, think about it in your head. We are pumping 3 sodium out of the cell and then bringing in 2 potassium. Each sodium has +1 charge and each potassium has +1 charge. So for each cycle, we are losing 3 positives and we are bringing in 2 positive. So my question to you is, if we let this happen for some time, ultimately, is it more positive outside or is it more positive inside?
More positive outside.
Which is it? Sorry, Shelley?
And more positive outside.
Outside. Okay. And Leah, did you?
That's the same thing.
Yeah. Does everybody understand that it's going to be more positive outside? Because in each cycle, you are like losing a positive to the ECF, right? So over time, over time, we are going to have more positive charge on the outside. Positive. Okay. Which means the inside is going to be more negative. Okay, so let's write that down. So if you measure the charge difference between the inside and the outside, we call that the membrane potential. Membrane potential. So at rest as in like the neuron is not transmitting a signal, at rest, the resting membrane potential, or we call that the resting potential, is approximately minus 70 millivolts. Just to help you picture what this is, you know your standard AA battery, right? Anybody know how much voltage that thing has? Any people, anyone pay attention to that? It says on the battery. I'm just curious if anyone knows. No? Yeah, exactly. 1.5 volt. Okay. You might know the square one, right? The rectangle one, that's like 9 volt, right? So voltage is a measurement of charge difference. In the context of the battery, it means like if you measure the positive and the negative terminal, there is a difference of 1.5 volts. Okay. So in the body, we don't say volts because that's like too much. It's gonna cause our cell to damage. We say millivolts. There are 1,000 millivolts in a volt, right? So if we measure the inside of the membrane compared to the outside, the difference would be minus 70 millivolts. Okay, so this is called a resting potential because the neuron is not transmitting a signal. Okay, and so it is correct to say that the resting potential is created by, is established by the sodium-potassium pump. Okay. So on that second piece of paper that I asked you to have, we're going to draw a graph. Okay. So big graph. That's the y-axis, that's the x-axis. This is going to be membrane potential, and it's measured in millivolts. And then over here, we have time, which is measured in milliseconds. Okay. So we're going to start right here at -70 millivolts. Put a line right there. Right. So this is what we call the resting potential. And I also want us to focus on the concentration of sodium and potassium and how they change at different parts of this graph. At resting potential, on the inside, we're always going to be talking about the inside, all right, unless otherwise specified. On the inside, what's the sodium level? Right? See if you remember. Is it high or low? Let's think about that for a second.
Low.
Low is correct, right? Because we keep on pumping it out, right? And then we would have high potassium here. Okay. So that's the setup right now. Any question about any of the stuff that we talk about so far? We are doing background information, number 1. If we don't have question, we'll move on to background information, number 2. All right. So still in background information, we're going to talk about the second piece now. Okay. We'll call this voltage, voltage sensitive, gated channels. So gated channels, you remember from, again, last semester, right? We talk about like facilitated diffusion, right? Where things pass through the membrane with the help of a membrane protein. So some of these membrane proteins, they are gated. Okay, there is a gate to them. Right? They either are closed or they're open. That's what gated means. And depending on the kind of channels we're talking about, they are usually sensitive to certain things that would cause them to open or cause them to close. In this case, they are sensitive to voltage. So the membrane voltage, the membrane potential will decide whether the channels are open or closed. Okay? There are other channels that are controlled by other things. Right? Some are controlled by pressure. Some are controlled by the presence of certain chemicals. Right? But in this case, they are controlled by voltage. So there are 2 types that I would like to talk about. And you probably guess what they are already, right? So there are the sodium gated channels. Think of them as like a door, okay? And this is closed. So this is a closed sodium channel. And when the voltage is correct, then they will open. So this is an opened sodium channel, right? And, needless to say, sodium channel will only allow sodium to pass through, right? So if this is the ECF and this is the cytoplasm, right? Which way is sodium gonna move when you open the door? Into the cytoplasm or out? We talked about this earlier, right? Hopefully your answer is that they're gonna come into the cytoplasm, right? Because you have so many red dots on the outside, and then, you know, very little on the inside. So they will come into the cytoplasm. All right. And so, you know, what voltage will they open at? Well, they will open at -60. And they will close at +30. All right. It's like, you know, store hours. Open at -60, close at +30. Similarly, we are going to have the blue ones. This is a closed potassium channel. This is an open potassium channel. And, you know, we have, let's say we have the ECF on the top and cytoplasm at the bottom. And again, when they are open, which way will the potassium go? Hopefully, you are thinking it will go out, because you have more potassium on the inside than outside, right? So which one does this, when does this open? Well, it's gonna be opening at +30. And it will close at -90. Okay. All right. Here's one last little thing to consider. Look at my hands, or my fists are like the channels. Okay, so this is closed, this is open. Right. So pretend this is sodium channels. We are what number are we sitting at right now? Like when we're not doing anything, when the neurons are not doing anything, what number are we sitting at? What's the membrane potential at this point? Right? -70, right? We said so at -70, which is lower than -60, all the sodium channels will be closed like that, right? Okay. So once we get to -60, and I'll tell you how we get there in a bit. Once we get to -60, they will open, okay? And then eventually they will close at 30. Right? Here's the thing to pay attention to. For both the sodium and the potassium channel, once they open and closes, they will enter a refractory period. During this refractory period, they will remain shut no matter what. Even if you reach the triggering voltage again, they will not respond. Okay. During the refractory period, the channels are resetting themselves. Okay. They must reset before they respond. Okay. So once these channels open and then close, they enter a refractory period. And this refractory period is important. We'll come back to it later. During which they won't open, even though the opening voltage is reached. Okay. So they need to reset themselves. It doesn't last very long. It's just a couple like milliseconds. And then it resets. Once it resets, it's able to do it again. Do you understand? Like as a loose analogy to help you remember this. Yeah, like I think of my kids. Like I hate taking them to the bathroom when we're outside. It's just like very difficult for me to take them to the bathroom when they're at the mall or something. Okay. So I always ask them before we leave the house, was like, make sure you go pee, okay? And then my daughter, because she knows I'm gonna ask her to pee, so sometimes she already pees, and she'll be like, but I just went, I can't. I was like, go again, just try. She said, I can't, I just went, right? So that's kind of like the refractory period, right? The channels open and closes, okay? It just opened, so it can't open again. It needs to reset. You need to wait a little bit, okay? No matter how hard you try to tell it to open again, it just can't. It just needs to reset itself, okay? I don't know if you guys make the connection. It's like that that example, that scenario. It's kind of an analogy for me to remember something like that. So are we good? Like that's all the background information, right? With the sodium potassium pump and with the voltage sensitive gate channels. If we are all good, then we're gonna move on to actually learn about the action potential and finish the graph. Really? No question? Fantastic. Okay. Let's talk about the action potential now. So let's just redraw some of the stuff. Right? I'm gonna draw like a super long one. A super long one like this. Oh, I have a question. When is the reset? Sorry, Min, you asked all these good questions, and I feel bad because every time you ask a question, I'm like, hold on to that thought. We're gonna come back to it. We will. It's just gonna reset a little bit, like a couple of milliseconds, and then it's gonna be good to go again. And when we finish the graph, I will tell you exactly like when the resetting is happening. It'll make much more sense there. Okay. If I don't answer your question by the end of the lecture, just remind me again, and then I'll be sure to address that. But excellent question. All right, so this is the inside, this is the outside, right? Okay, now I'm just trying to make this simple so we don't over clutter our diagram. We know that we have a bunch of sodium potassium channels here, right? The purple balls, but we're not going to draw that, okay? Because I don't want to make the diagram too busy, okay? But we will draw these things. Okay, so if you have been with me since the beginning, you will know what these are, right? Hopefully you would know what these are. These are sodium channels. And they're closed right now because we are at rest. Okay, minus 70 millivolts. And then we would have all the potassium channels. All the potassium channels. You definitely will have to reorganize your notes afterwards, because the way you draw it should be in a way that helps you understand what's going on. Okay, so again, we have all these red dots on the outside, right? We'll have some on the inside. And then it's the opposite for the blue ones. There we go. Okay. So listen, all these red dots, they really want to come in from high to low, right? But the doors are all shut. We are sitting at minus 70. None of them can come in at the moment, right? Okay. Think of them as those shoppers, Black Friday. They're all lining up outside of the store, ready to flood the store as soon as the doors are open. Okay. And then the blue ones, they are like the Walmart employees inside the store, ready to get out. Okay, but they can't. The doors are also closed. Okay. So that's the setup right now. Okay. Then something happens, okay? Like a stimulus arrives. So I'm gonna draw a new one here. This is in the other diagram. It's a similar information, okay, but pay attention to what the difference is. So what happens here is we're gonna have a stimulus arrive, some kind of trigger. Okay, maybe it's the flashlight in your eye, maybe you touch a hot surface, whatever the stimulus is. Okay, so the stimulus arrives, represented here by a lightning bolt stimulus. And that is gonna push the membrane potential higher. We started at -70, right? When the stimulus arrives, it will go a little bit higher. Okay, -65, -63, -62. Eventually, you hit the magic number of -60. Why is -60 a magic number? Well, we said that at -60, that's when the sodium channels are going to open. Right? So when the threshold reaches -60, we will open these sodium channels. Okay, so that's going to open up the sodium channels. Oops, wrong color. Open them up, open them up. Now the axon is really, really long, right? You remember. So the stimulus, the signal is only at the beginning portion here. So only the sodium channels over here will open. The rest of them is still closed. Okay, the signal, the stimulus hasn't reached that point yet. Okay, so the stimulus will push the membrane potential towards the -60, and when you do hit the -60, the sodium channels will open. And as soon as they open, what happens to the red dots? Where do they go? Well, I've been repeating it the whole class, so hopefully you know that they will be coming in, right? They'll be coming in. Yeah, so you have all these sodium flooding the store, right? Like I said, going into the cytoplasm. Further down though, further down the axon, no changes occur. It's still same as before because the doors haven't been opened yet at those locations. Okay, now if all the dots are confusing you, let's put the descriptor there. Okay, this is gonna be high sodium on the inside now, right? You understand? Because they all came in. Low sodium on the outside. Further down the axon, where the action is not going on yet, doors are not open, sodium channels are not open, right? We will still have high sodium on the outside and low sodium on the inside. Do you understand? Help me out here, guys. What number should I put here for the membrane potential further down the axon?
-7.
Yes, -70, because nothing happened yet, right? It's still the same. Okay. Over here, though, that's a different story. Okay. I'm gonna give you a moment to, copy whatever you need, and then we're gonna switch over to the graph. And then I'm gonna show you. All right. Lightning bolt. Stimulus arrives, and it will push the membrane potential towards -60, right? How that happens, I will show you in a second. Okay, just trust me, it's gonna push it towards -60. Okay, so that -60 right here, that is our threshold potential. Okay, it's called the threshold because once you get past this point, it's a point of no return. All things gonna happen. Okay, so at this point, right here, at -60, sodium channels open. As soon as the sodium channels open, all the sodium is gonna flood inside the cytoplasm. So please tell me, with all the sodium coming in, what's going to happen to the membrane potential? Is it going to get more positive? So A, more positive. B, no change. Or C, more negative. And we're talking about the inside, right? Inside is always our point of reference. Okay, so please let me know. My question is, as soon as the sodium channels, right, what's going to happen to the membrane potential? Is it going to be more positive, no change, or more negative? Okay, beautiful. We have overwhelming people saying it's going to become positive. Of course, it's going to become positive because all the sodiums are coming in and all the sodium carries a positive charge, right? Right. So with the sodium influx, we call it, the graph is going to go up. It's going to be more and more positive. In fact, it's going to go past 0, and then it's going to keep on going up and up and up and up. Okay, so sodium will keep on coming in until we close those doors. Okay, and what voltage would that be? When would the sodium channels be closed? Right. At what number? Do you guys remember?
30.
Yeah, if you look back at the notes, right, that we took in the background information, we said it's going to close at positive 30. Okay, so at positive 30, couple of things are going to happen. Right, so I'm going to put them down here. The first thing that happens is sodium channels that were open will now close. Okay, more stuff to come later. Now this part here, this is called the depolarization phase. Depolarization, and it's caused by sodium influx. Okay, in the context of what we're talking about, polarization means like more negative on the inside. That's what it means. So when you are depolarizing it, you are making it less negative. Right? And what's making it less negative? It's the sodium coming in, right? Sodium influx. All right, so let's take a look at our diagram again. Now we have been focusing on the sodium and completely ignoring the potassium. Let's think about the potassium. What's the descriptor for potassium on the inside at this point? Should I put high or should I put low? This was the before stay, and now after the sodium channel opens, what are we going to say about the potassium? Is it high or low?
Low.
And why is that?
Because the potassium, there's more potassium, what is it, the sodium than the potassium?
Okay, interesting. Ignore the sodium for now. Okay, just focus on the blue dots. Do we have more blue dots on the inside or outside after the sodium channel opens?
Unchanged.
It's unchanged. That's right. Because the potassium, as much as they want to leave, with all the sodium coming in, they really want to leave, right? They can't. How come they cannot leave? Because the potassium channels are still closed, right? Okay, so if we draw the potassium channels here, right, they are still closed, right? Still closed. So just like before, it's still very high. Does that make sense, Sashana? Does it, like,
Yeah.
yeah, okay. So this is going to be high still, and then this is going to be low. So you might want to make a note here and say why it is still high and low. So despite the fact that all these sodium are coming in, since the potassium channels are still closed, the potassium has nowhere to go, right? All right. let me explain to you what happens next, rather than drawing a diagram. Okay, try to follow. So we are at the peak of our graph right now, right? If you think about the graph we drew, we went all the way up. We just went through the depolarization phase, and we go all the way up to 30. And at this point, we shut the sodium channels. They enter the refractory period. Okay? They just open and close. They can't open again. They can't go just yet. They have to reset, right? And now we're sitting at positive 30. So sodium channel closes. What's the second thing that happens? Well, if you look at the notes that we wrote, positive 30 is the voltage for the potassium channels to open. Right? So now is the chance for the potassium to leave. Okay. But there's more to the story here. Okay? So I'm going to put it over here. At positive 30 millivolts, potassium channels open. Okay. And then there are two driving forces. That push potassium out. Okay. First one is the easy one. Okay, concentration gradient. We already established that things naturally goes from high concentration to low concentration. They wanted to leave on their own anyway, right? The blue dots, if we go back to our previous diagram here, right? We have a lot of blue on the inside, so naturally they would want to move out of the cytoplasm. Okay, so that's one driving force. The second driving force has to do with the sodium. Right? The sodium all came in because of the depolarization, and now the inside of the cytoplasm is super positive. Right? It's very positive, and positive charges, they don't like to be next to each other. Right? Light charges repel. So not only does the potassium want to go down its concentration gradient, it also wants to get away from all these positive charges that are coming in. So a second driving force that would really push the sodium out, right, is the electrostatic repulsion. Right? Which means like charges repel, right? So with these two driving forces pushing the potassium out, right, then you're going to be losing positive charges, which means your membrane potential will now become more and more negative again, right? So if we have the graph, after opening this potassium channel, it will come back down. Okay, and again, we have two driving forces, so you will have a lot of potassium leaving, so much so that when this is all said and done, you would be all the way down to minus 90. Okay, so this here, this phase, this falling phase is called repolarization. Remember, polarization in the context of what we're talking about means having a negative charge on the inside, right? So now we are re-establishing, recreating the negative charge. And that is caused by potassium efflux, okay, leaving. All right, so let's go back to that question you were asking, right, Min, about the refractory period. Take a look here, guys. As the graph comes back down, we're gonna hit the threshold again of minus 60, right? Okay. So does that mean the sodium channel will open again at this point? Well, the answer is no, because the sodium channel already open and closes. So even though we reach minus 60 again at this point, they will not open again. not for this time anyways, because they're in the refractory period, right? And it's important to have that. Otherwise, if you open the sodium channels here again, then you will have the sodium moving out, and then you would just have like a fluctuation of signal going all over the place, right? And that's gonna cause you to not be able to transmit the signal properly. Right? So that's why we have to have the refractory period, and it doesn't have to be very long. It just has to be longer than this duration. Okay, so we just have to make sure that when the graph comes back down, we're not going to open those sodium channels. So I hope that answers your question. Okay, so at this point, right, at minus 90 over here, the potassium channels will close now. And this point, it's like more negative than resting. So we call this hyperpolarization. Hyperpolarization. Too negative, that's what it means. All right, so let's just put some descriptor here for the concentration again, right? At the peak here, because the sodium came in, so we're now gonna have high sodium. And again, just want to remind you, we are talking about the inside, right? Relative to the outside. It's always gonna be inside unless otherwise specified. And what about potassium here? What did we say again for potassium? Was it high or was it low? Hopefully you're thinking high. They didn't go anywhere, right? And then afterwards, after the efflux, after the repolarization, okay, at this point, at minus 90, at hyperpolarization, what should we put for the sodium?
Unchanged.
Okay, so is it high or is it low?
High.
High. Does anyone think it's low? Or do we agree it's high? Yeah, they didn't go anywhere, right? They came in and then the door shuts, right? So you trap all the shoppers in the store. They can't go anywhere. They have to stay inside. And then the potassium would be low, right? Because they all left, right? So at hyperpolarization, we have two problems here. Right? I guess I'm just going to write a note here. And again, I encourage you to reorganize your notes after class. Okay, so over here at hyperpolarization, we got two problems, two situations here. Number one, we are more negative than resting potential. That's a problem because, you know, it would, in order for you to reach the threshold again, you would need to, you know, have a difference of 30 millivolts. Okay, so that's like too high, right? It's hard to get to that point. So we're more negative than resting potential. And problem number two, if you pay attention to the ion distribution, it's completely reverse compared to what it was supposed to be at rest, right? So ion distribution is backwards. It's reverse. So at this point, the neurons can't do anything. It's not going to be able to transmit another signal. Okay. So what you must do is we will have to reset it back to minus 70. Right? And to reset it, that will be the job of what? Who's gonna come in and reset our ion distribution and bring it back to minus 70? The pump, that's right. The sodium potassium pump will reset the resting potential. Okay, and then the neuron will be able to transmit another signal. Okay. So this part right here, this part where the neuron membrane potential temporarily switched to positive, that's what is referred to as the action potential. Okay, so this right here, temporary flip in membrane potential. Right? That is what we mean when we say action potential. Okay. Remember, potential is just like a difference in charge, right? So that's where the action is happening, cause a flip. But the flip is not permanent, right? As soon as you open the potassium channels, you're gonna, undo the flip. Okay, I just have a couple more little things to tell you about before we get going. But seriously, like anything you want to ask at all that requires clarification about any of these things?
I just don't understand why when the membrane potential reached -60, the sodium channel will open.
Okay, you don't understand why the sodium channel will open at -60? Is that the question?
Yeah.
Okay. The better question is, how did we get from -70 to -60? Okay. Now don't worry about why it opens at 60, right? That's the way that the channels are designed, right? Like, whoever created us, the sodium channels, it's designed to open at -60. That's just a trigger for it, right? Okay. But how exactly did we go from -70 to -60? That's the thing that we're going to be focusing on. Okay, so let's go back to here. Now I know this diagram is not really complete, and I don't want to draw too many of these. Is already confusing as it is. I'll post a video of like only of me explaining only the action potential portion, so you don't have to skip through the entire lecture to find it. I have a clean video of just me talking about this stuff for like 20 minutes. You can watch that if you need to. So right, the sodium came in already, and then sure, the next thing that's going to happen, the potassium is going to leave, right? Okay, now remember, what's also going to happen is that the sodium channels will close after the sodium comes in, right? Okay, so I'm just gonna show that here. It's really hard to draw all these changes on a static diagram, but we're gonna close these red doors. Close them. There. Okay, so we are at the peak right now at +30. Okay, we can close all these doors. Potassium channel is going to open next, sure, but we're not going to focus on that. We're going to focus on the sodium. Okay, so we have a lot of sodium here, and don't forget, at this moment we also have a lot of potassium. So the sodium would actually want to get away from them as well, because like I said, positive charges repel each other, right? So the sodium wants to go back out, but it can't. The doors are closed. So there's only one way to go. Which way is that? Right? Take a look at the diagram. And the only way to move now is forward, right? Because further down the axon, where the action hasn't been happening yet, it's still sitting at -70, and we have low sodium, right? So some of the sodium is going to creep forward, right? And then listen, we were sitting at -70 for this section, but as the sodium move forward, this is going to become a higher number, right? This is going to be like -65, -63, -62, and then eventually we're going to get to -60, and that's the threshold potential. Then the sodium channels of this section will open, and then all the sodium would come in, and then it will cause the next section to experience the flip, right? And then the next section will experience the flip. Do you understand that? Okay. So over here, the third thing that's going to happen at the peak here is that sodium channels of the current section will close, but sodium channels further down the axon will open. Okay? That's how the signal is going to be propagated. Okay, let's put all these together in a series of diagrams that hopefully will create a clearer picture for you. You don't have to draw this. You can just like watch me doodle, and then you can copy it at a later time. Okay, so this is the neuron with the axon, and let's say we're at rest. So minus 70, minus 70, minus 70, all the way, right? Okay. So then, let's say we have some signals arriving. Okay, there are some inhibitory signals coming in at the dendrites, and then there are some excitatory signals that are coming in. Okay, but there are more excitatory signals than inhibitory signals, and so that's going to cause a signal to be transmitted down the axon. Okay, so here we go. When we decided to allow the signal to pass on, right, we will have the lightning bolt. We'll talk more about that next class of how that actually happens. But the lightning bolt is going to push this towards the threshold, right? So now it's going to be negative 60. The rest of them, though, is still going to be minus 70, right? Don't copy this down. You'll miss what I'm saying, okay? Just focus on what I'm saying and then try to, reorganize it yourself afterwards. Do you understand why this is minus 60, right? Because of the lightning bolt, right? Because of the stimulus. Okay. So then what happens next? What happens next is, because we've crossed the threshold, then the sodium channels will open. When the sodium channels open, we will experience depolarization. It will cause this portion to become positive 30. Okay. And then what happens is, because of the sodium coming in, and then you will close the door, it will push the next section towards minus 60, right? Because some of the sodium creeps forward afterwards, right? But the rest will still be at minus 70. Okay, so this part here flips to 30. So we'll put a little light bulb here, ding, to light it up. Then what happens next? And we're going to draw another neuron. All right, so after we reach the peak of 30, sodium channel closes, potassium channel opens. After potassium channels open, all the potassium will be leaving, and then it will cause this section to plummet down to minus 90. And then the next section, because it was reaching the threshold, right? So now this section is going to have the sodium channel open, and now it's going to get pushed towards minus 30. And once it reaches minus 30, some of the sodium will creep forward and it will push the next section towards the threshold, while the rest of them is going to be at minus 70, right? So the next portion is going to experience the flip, right? The temporary flip, the action potential. So ding, that lights up. Just going to draw one more. And then what happens is, this was sitting at minus 90. We are at a hyperpolarized state. We are way too negative. Our ion distribution is all wrong. So the sodium potassium channel will come in and it will redistribute the ion and then reset it back to minus 70. Okay, and as this is happening, this will plumb into minus 90, hyperpolarized. And then the next part will experience the action potential, right? Causing the next part to go into the threshold, and then everything else remains the same. Okay, so now, ding. Do you understand? Okay, so if we ignore the numbers, if we just focus on the neurons, okay, let me see if I can do this. So it's like ding, right? The action potential. So this lights up, and then cause the next part to ding, to light up, and then it will just keep on passing down and passing down. This is terrible animation, but hopefully you get what I'm saying, right? It's just the 30 that's get flip, 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 right? One part causes the next part to flip, and then the next part to flip, and then the next part to flip, until the signal gets all the way to the axon terminal, right? So that's how it's being passed down in the axon. Yeah, thank you. That is an excellent analogy. It's like the human wave, right? In the stadium, right? Okay, that progressively get propagated down the axon. I have a private message. Can you explain again how the -90? Yeah, sure. Okay, that's it. So -90 is right here, right? That's like when it's like hyperpolarized. It's too negative. The ions is all wrong, right? We have the sodium on the inside, and then we have the potassium that's outside. So we need to reset it, and that's the job of the sodium potassium pump, right? Sodium potassium pump will take all these extra sodium that's not supposed to be inside and send them back out, right? 3 at a time, and then we'll be bringing in 2 potassium to reset the resting potential. Does that answer your question? All right, great. So yeah, so eventually the signal will get to the very end, and then next class we will see how the signal jumps from one neuron to the other neuron. Okay, so what I want you guys to do is to draw your own graph, this graph, okay, and then put in some detail on the graph, and then you should use this graph as a help sheet on the quiz, right? Okay, it's hard to keep track of all the information, perhaps, to memorize it. So it's good to have a sheet with key information on it. Pay attention to the ion distribution, what's coming in, what's going out, and I want you to create that sheet between now and Wednesday. Okay, and then we will start our class with some of these poll questions, and then we'll talk about how signal goes from one cell to another. Okay, so I'll post a video of this recording, obviously, and then I'll post another one, like one of the older ones that I've created just for the action potential, in case you need a second dose of that. Okay, the class notes though, I think I'll wait. Oh, okay, maybe I shouldn't wait. Okay, I'll just post it already. Okay, and then when we add more next on Wednesday, we'll just post another set. Okay, that's it. Thank you. See you all on Wednesday.
Professor.
Yeah.
You still haven't talked about why when the stimulus make the -70 to -60. You're going to talk about this next class?
No, I did already, right? But I'll go through it again. That's okay. Okay, look. This was sitting at -70, right? Oh, feel free to go, okay? If you guys are done and you understand, feel free to go, okay? So this was sitting at -70, right? Okay, so the sodium that just came in for this first part here, they have nowhere to go. They can't go back out because the sodium channels are all closed, right?
Okay.
So the only place for them to go is to go forward, because further down the axon, the sodium concentration is still low, right?
Oh, I understand this, but my question is how it happens at the first place.
Oh, like this one, right? Like
this one, yeah.
Yes. Okay, so that's the next class, yeah.
Okay, next class. Okay.
Okay.
Thank you.
No problem.